So we now come to the end of the chapter respiratory system, because now we have got certain only experiments balanced in the chapter. So there are in all around 5 experiments, very simple and small experiments which are there, which is going to demonstrate that what are the important things. How means carbon dioxide required, oxygen is given out, taken in, carbon dioxide given out. So ये सब easy things है. In fact, very rarely they ask questions based on the experiments of respiratory system in humans. Normally respiration का अगर कोई भी experiment आता है तो normally वो plants का आता है. Humans का थोड़ा chances कम है. Anyway, we will go for the different experiments one by one. So first, to demonstrate the water is lost during breathing. So first thing is that you want to prove that the air which is going to be expelled out during breathing is going to contain water vapor. So for that, it's a very simple thing. Gently breathe upon a cold surface such as a piece of glass or slate. The water droplets appearing on the surface prove the presence of moisture in the expired air. So you always see a glass के ऊपर भी now, but when you see people doing it, कि अगर चश्मा को साफ करना है तो they will ऐसे करके उन लोग breathe out करते हैं वहाँ पे, तो they will form a fog, so like a fog जैसा बनने लगता है उसके ऊपर on the glass, and then they rub it. So they, instead of just to make it slightly wet, they just exhale out over there. So that's how you can identify that water vapor was present in the expired air. Second is to demonstrate that carbon dioxide is given out in breathing. So for this, the simple thing is that you are going to take 2 flasks. Now the 2 flasks will be such that you are going to have in one the lime water, and another, both will have lime water, and the air is going to be, and then you are going to suck the air and you are going to let it out. So when you are sucking the air, it is passing through the lime water. You will find that the lime water turns milky, proving that the air which was coming inside was having carbon dioxide, so which has got absorbed in the lime water over there. So what we have taken in is free from carbon dioxide now, and when we expire out, at that time you put a clip so it doesn't go on the other side, and you pass it from this side, and at that time when it passes through the air, you will find that it will turn, lime water turn milky. In fact, initial case में lime water turn milky नहीं होगा, because the carbon dioxide content in the air is only 0.04%. So yeah, it will not turn milky, whereas the one which is expired out will have milky. So you can see that the air did not have carbon dioxide initially, but after exhalation, it was having it. So that's a setup by apparatus as shown in the figure 14.9. Clip C is open and clip D is closed. Clip C is the one where you are breathing in. So for breathing in, यहाँ से जो breathing करते हैं, तो clip AC जो है उसको open रखा है, clip D is closed. Air is sucked in by the mouth through the tube at the center. Atmospheric air rushes in the flask A, bubbling through the lime water. Next, clip C is closed and clip D is open, and air is blown through same central tube. This time, the air is forced into the flask, bubbling through its lime water. The process is repeated about 10 times. The lime water in flask B turns milky much faster than the flask A. This proves that the expired air contains more carbon dioxide than the inspired air. So you're just going to keep on doing the thing. You're breathing in, breathing out, and breathing out. वैसे करके हम लोग करते जाएंगे, और ये उसके हिसाब से the A flask, which is the air is taken in, does not become milky, whereas the B flask, which is exhaled out, is going to turn milky, proving that whatever air we are given out contains carbon dioxide in it. We move on to the third experiment. To demonstrate the action of the diaphragm during the breathing. So here in this case, what we are doing is we are taking a bell jar, and this bell jar has and you are going to place over here a tube like this, to which you are going to keep in the balloons over here, okay, and this over here you are going to put a stretched rubber band, okay, so stretched rubber sheet will be there. The moment you stretch this thing, so you stretch this balloon, this particular rubber sheet, you will find that this automatically gets inflated, because when I push this downwards, the air inside has got the volume inside and increased due to the pushing over here. So this gets a low pressure. Because there is a low pressure, the air enters from here to make this inflate. So, and similarly, when you push this thing inside over here, this is going to deflate again. So this is how the lungs are going to perform. So you can consider this balloons to be our lungs, and this is the diaphragm. So the diaphragm working is being understood by this particular experiment. Set up the experiment as shown in the figure 14.10. The rubber sheet tied around the bottom of the edge of the bell jar represents the diaphragm. When the sheet is pulled down, volume is increased, pressure inside the bell jar lowered, and the rubber balloon are expanded by the air rushing in through the tube at the top. When the sheet is pushed downward upwards, volume is decreased, pressure inside the jar increases, and the balloon collapses due to the air rushing out. The balloon represents the two lungs. So that was the experiment to show the diaphragm movement. The fourth is to measure the expired air. So for this, what you are having is, set up the experiment as shown in the figure. Fill your chest with air to the maximum. So you are going to take a deep breath, okay, and then you are going to take a bottle, and in the bottle you are going to fill up water up to this point. There will be this tube like this, and another tube will be over here like this, which is going to collect the water over here. So the water is going to get collected here. So you are going to breathe out like this inside. So when you are breathing it out, so this is completely sealed. So when you are breathing it out, there will be pressure generated over here, due to which the water is going to push upwards over here. Okay. So fill your chest with the air in the maximum and then blow out through the short tube, expelling out much air as you can. The water expelled from the other tube when measured gives the volume of the air exhaled. So whatever is the water which is going to be collected over here is going to be the volume of the exhaled air. The fifth and the last experiment is to show that oxygen is taken in by animals during respiration. Use a small insect such as a cockroach or a rat in this experiment. Take two conical flasks A and B. Place a live cockroach in one flask A and a dead cockroach that has been soaked in formalin to prevent decay in the other flask B. So you are going to take two flasks A and B. You are going to take two cockroach, one dead and one alive. The dead one, you will have to put it in formalin and then use it so that it is not getting decayed, because if it starts decaying, then the bacteria which are there, which are causing the decay, they may respire and show you the wrong results. The flask with the dead cockroach acts as a control. So you know that there should not be any change in the flask B which has got the dead cockroach. Fit a rubber cork in the mouth of each flask and make sure that the apparatus is airtight. Leave the flask for a few hours, after which introduce a small burning candle into each flask as shown in the figure. So what you are doing is, you are taking this flask, दोनों को ये अंदर कॉकरोच है, हम लोग ने उसके ऊपर लिड लगा दिया है, okay? Now you know that the one which is having a live cockroach inside will be breathing continuously. So the air inside that particular flask will keep on decreasing, whereas the one which has got the dead cockroach, there the air is going to remain intact. So after some few hours, you are going to open the lid and introduce a burning candle inside that thing. So when you introduce the burning candle inside, you will find that the conical flask which contain the live cockroach, there the candle extinguishes immediately because there is a lack of oxygen over there. Whereas the one which has the dead cockroach, there the candle keeps on glowing because the oxygen was not utilized by the dead cockroach, obviously. So leave the flask for a few hours, after which introduce a small burning candle into each flask as shown in the figure 14.12. Immediately after introducing the candle, close the mouth of the flask tightly. Note the time taken for the candle flame to go out. You will find that the flame goes out faster in flask A containing the living animal, Since oxygen supports burning, the flame of the candle goes out faster because the living cockroach has also consumes oxygen for respiration. So this is how you can prove that the oxygen is going to be required or taken in by the animals. We now come to the final thing in the chapter, and that is a comparison of respiration in plants and animals. So first of all is the similarity between the plants and animals respiration. The first is cellular respiration produces energy stored in ATP inside mitochondria. So in both the cases, both in case of plants as well as animals, the mitochondria is going to produce the energy in the form of ATP. And secondly, aerobic respiration forms carbon dioxide and water. The similarity will be that in case of aerobic respiration, there will be a formation of carbon dioxide and water. So differences now, no external ventilation, breathing movement. So there is no external ventilation. In case of the plants, there is no external ventilation or breathing movement going to be there. Whereas animal respiration, external ventilation movements, except in extremely small animals like amoeba or hydra, will not be there. Other is no special gas transport system, actually direct diffusion from airspace or from cell to cell. Whereas in case of animals, blood system, transport system required for the oxygen movement. So blood system transports the oxygen. In plants, anaerobic respiration forms ethanol and carbon dioxide, whereas in animals, anaerobic respiration forms lactic acid only and no carbon dioxide. In plants, green plants produce little detachable heat, whereas animals produce considerable detachable heat. Green plants have additional oxygen source from photosynthesis. Yes, in green plants, the oxygen can be available even from photosynthesis, whereas aerobic respiration animals have only air as a source of oxygen during breathing. Finally, plant respiration rate is low, whereas animal respiration rate is high. So this was the end of the chapter respiratory system. So quite an easy chapter. Mainly what we have to focus is about the parts of the lungs, the inner wall, the outer wall, the alveoli, the alveolus gaseous transport. These are the main important parts in this particular chapter. Very important because they are the one which can normally be asked. So in fact, this figure is something which is asked a lot of times. Questions based on this are asked a lot of times. They give this figure and they ask questions based on that. So overall, yes, a very easy chapter as such, right? Okay, so that's the end of respiratory system. Thank you.